Hello, my name is Lawrence Guessman. I'm a fourth year medical student from Imperial College London, and today I'll be presenting my project investigating the mechanisms of resistance to CCS by multiple myeloma. So what is multiple myeloma? It is a malignant proliferation of plasma cells. It is the second most common blood cancer, and it is considered a disease of old age with median diagnosis being 70 years old. Corticosteroids, immunomodulatory drugs such as pomalidomide and proteasome inhibitors are the mainstay treatments. However, as patients are on these therapies for life, drug resistance becomes common. This necessitates the development of new therapies for myeloma and also creates a burden on the NHS with unplanned hospital visits being common and many patients reaching as many as 7 lines of therapy. IRF4 and MYC are two important oncogenes for myeloma survival. IKZF1 and 3 are two transcription factors that drive IRF4 and MYC expression. That is the target of immunomodulatory drugs such as pomalidomide, which bridge the gap between IKZF1 and 3 and cereblon, an enzyme that degrades them, leading to suppression of IRF4 and MYC. However, as I mentioned, drug resistance is common. As such, an alternative target, P300 and CBP, is important. These are two paralogous cofactors that drive IRF4 and MYC expression, which I'll refer to from now on as P300 collectively. CCS1477 is a novel therapy which is in phase 1 and 2A trials at the moment. It targets the bromo domain of P300 to suppress IRF4 and MYC. And we hypothesize that as this drug is rolled out into the clinic, CCS resistance will arise. As such, my primary question is, how might myeloma become resistant to CCS in the clinic? I answered this by looking at CCS-resistant myeloma cell lines developed by the lab and treating them with different inhibitors to investigate effects on growth, protein, and RNA levels. The first question was, how is gene expression affected by CCS resistance? Using RNA sequence analysis, we found large-scale downregulation and upregulation of genes. We think these were mostly transcription factors, which made us want to look at IRF4 and MYC, which are two important myeloma transcription factors. In sensitive cells, we found that in CCS compared to DMSO control on Western blots, that MYC and IRF4 were reduced in CCS. However, in resistant cells, they were maintained, which speaks to their importance to the CCS resistance mechanism. If cells are resistant to CCS, could they be resistant to other P300 inhibitors as well? As such, we looked at A485, which targets the hat domain of P300 to suppress IRF4 and MYC. If A485 works while CCS doesn't due to CCS resistance, this might point to the source of the resistance being in the bromo domain that's targeted by CCS. As such, this is a growth assay of cells counted at 72 hours, which showed that the cells grown in DMSO, CCS, and A485, CCS and A485 cells that were sensitive to CCS grew slower. However, CCS-resistant cells maintained growth in CCS and A485, which shows that CCS resistance confers resistance to A485. H3K27AC is an indicator of whether or not CCS and A485 are having an effect within the cell. When we looked at a Western blot of this protein in DMSO, CCS, and A485, found it was reduced in the sensitive cells and in the resistant cells too. As such, this makes the likelihood of a drug efflux mechanism less likely. What are the effects of pomalidomide on CCS-resistant cells? So we know that CCS resistance confers resistance to A485. This made me wonder, could it confer resistance to immunomodulatory drugs as well, such as pomalidomide? As such, I grew cells in DMSO, pomalidomide, CCS, and combination. In sensitive cells, as expected, there was reduced growth in the treatment groups. However, when we looked at CCS and pomalidomide grown cells, it was found that they both maintained growth in the resistant cell group, which shows that CCS resistance also confers resistance to immunomodulatory drugs. This raises the question of a common resistance mechanism between CCS and IMiDs. As such, I looked at IKZF3, the target of pomalidomide, and I found that it was reduced in sensitive cells and reduced in resistant cells, again reducing the likelihood of a drug efflux mechanism. I also found that IRF4 was again maintained to pomalidomide and CCS and combination, showing its importance in all of these resistance mechanisms. To summarize, CCS resistance confers IMiD resistance and A485 resistance. I identified IRF4 as a target for future research, and I would like to overexpress IRF4 in CCS sensitive cells to see if they become CCS resistant, and knock down IRF4 in CCS resistant cells to see if they become CCS sensitive. Finally, I think it's important to note that IRF4 is an important target for future therapies, as given its importance in myeloma survival in resistance. Some strengths were that the cells were human-derived. However, I could not recapitulate the tissue microenvironment that's important for these myeloma cells. And as such, I would like to recreate my model in more in vivo systems in the future. Problems I encountered included the fact that it was trickier than I expected to load Western blots, and also interpreting results is not as straightforward as I expected it to be. On reflection, I found that organizing my experiments using a calendar was helpful because it helped me keep track of what was going on. I found there's a steep learning curve for practical skills in the lab. It was difficult to balance my part-time job with my lab work. Again, organization helped with that, but working as part of a team helped with all of the above. And these are all skills that I'll take forward going with me into my career in medicine. I found that medicine is a lot like a cake. It has lots of ingredients, layers to it. It takes a team to make sometimes. However, it's generally good to those who have it. And my future advice would be, have an open mind, take care of yourself, and communicate with your supervisor for future students doing the BSc. And that's all. Thank you for listening.